Hello class, welcome to the module 1 of your molecular biology. For today's lecture, we will be discussing the introduction to your molecular biology. Presenting you here today, I am Ma'am Christina Matipa. So for your learning objectives or your learning outcomes, at the end of this session, you students must be able to, number 1, discuss the history and beginnings of molecular biology, and number 2, define the terms usually encountered in molecular biology. But first, let us define your molecular biology or the concepts underlying molecular biology. Number 1, it allows a scientist to investigate, manipulate, or study the composition, structure, and interactions of cells of an organism at molecular level. Usually, our molecular biology, it commonly deals with macromolecules. And when we say macromolecules, ano bang mga examples ng macromolecules natin? It is your nucleic acid. And when we say nucleic acid, there are two things that comes to our mind, and that is your DNA and RNA. So for molecular biology, we will be dealing with DNA and RNA for our journey dito sa ating molecular biology subject for our second semester. And your molecular biology is a specialized science that involves 3 branches of science, naming your genetics, biochemistry, and cell biology. Lastly, your molecular biology can be applied to these following fields. Number 1, it is used to diagnose diseases. So molecular biology, matagal na po talagang uso yan at ginagamit sa ating field of science, but it became more familiar to us nung nauso yung pandemic because the gold standard or the principle that is used to diagnose the COVID-19 is polymerase chain reaction, which is a principle in molecular biology. Next, it can also be applied to research and forensic analysis sa mga crime laboratories natin. Ginagamit po si molecular biology. It can also be used for gene therapy and paternity testing. Ang sabi po natin sa molecular biology, commonly, ginagamit natin at pinag-aaralan natin si DNA and RNA. So to give you a quick overview of the DNA, i-discuss natin siya sa next following slides. But to give you a more comprehensive discussion of your deoxyribonucleic acid or your DNA, i-discuss natin siya sa inyong module 2. Okay, so ito quick overview lang. Your DNA or your deoxyribonucleic acid, it carries the genetic instructions for almost every living thing. It is said to carry the genetic information, or its responsibility or its job is to code genetic information, yung hereditary material coming from our parents and kanununuan pa natin. Nandun po siya sa DNA. Okay, let us just read the description. It is normally occurs as a double helix, pakitandaan po, comprising 2 strands that are held together by hydrogen bonds between pairs of complementary nitrogenous bases. Okay, pakitandaan. I-discuss natin yan sa next illustration natin. Next, DNA are molecules that are polymers of nucleotide repeat units that consist of 1-4 types of nitrogenous base plus a sugar plus a phosphate. So pakitandaan po ito kasi i-discuss natin yan sa susunod nating illustration. So saan ba nanggaling ang ating DNA or saan siya nakalocate? It is located at the cell. It is located particularly in the nucleus. Which part of the cell? Particularly sa nucleus. Sa nucleus, nandoon yung inyong chromosome. Okay, sa chromosome, doon naka-embed ang ating mga DNA. Next, what comprises your DNA? Ano ba yung structure ng inyong DNA? So basically, your DNA is composed of these 3 following structures. Okay? Nucleotides. Nucleotides, ang tawag po sa kanila, sila yung building blocks of our DNA. Pag pinag-usapan naman si protein, ang building blocks nila si amino acid, whereas for your DNA, the building blocks will be nucleotides. Okay? So pag pinagsama-sama mo itong si phosphate, si sugar, and then nitrogenous base, mari meron ka ng helix dyan. Ang tawag dito sa helix na to ay isang nucleotide. Okay? So ang primary structure po ng ating DNA, it comprises of phosphate, your sugar, and then nitrogenous bases. Ano po ba ang sugar ni DNA? Ang tawag po doon ay deoxyribose. That is the sugar for your DNA. Deoxyribose. And then, ang mga nitrogenous bases naman niya, meron po tayong adenine, guanine, cytosine, and thymine. So, to discuss this much further, sa module 2 natin yan i-discuss. Bakit? Pero pakitandaan ng tatlong structure na nagko-comprise for our DNA, it is comprises of phosphate, a sugar, which is deoxyribose, and nitrogenous bases, which are adenine, guanine, cytosine, and then your thymine. So, kung babalikan lang natin yung description dun kanina, ito pong blue na to, ayan, two strands siya kung pansin nyo, parang ribbon. Ang nagko-comprise po dito sa ribbon na to is your phosphate plus sugar. Sila yung backbone of your DNA. Ito namang mga ladder na napansin nyo, base pair ang tawag dyan. Sa base pair na yan, nandyan yung mga nitrogenous bases natin, which are your adenine plus thymine, sila yung magka-partner, si guanine and cytosine, sila naman yung magka-partner para mabuo yung inyong base pair. Okay, so ang structure ng ating DNA ay it comprises of these three structures. So moving forward, these are the common molecular biology techniques. As you can notice sa pin-repair kong slides, meron tayong mga nabanggit na few molecular biology techniques, but we will discuss or we will elaborate those who are more commonly used in the laboratory these days. So, number one is your electrophoresis. I know you're very much familiar with electrophoresis because it is discussed in your analytical chemistry. And electrophoresis is a technique that is commonly used in laboratories to separate charged molecules like DNA, RNA, and proteins according to their size and charge as well. Madaming types or madaming examples or types ng medium that are being utilized for electrophoresis, but for molecular biology, it is the gel electrophoresis that is the one that has been commonly used or widely used for molecular biology, gel electrophoresis po. So, how does gel electrophoresis in microbiology works? In molecular biology works? For example, you have a sample. Meron kang sample cup or meron kang gustong i-determine or meron kang analyte dyan. And then the principle of gel electrophoresis is that itong sample mo, it contains mixture of DNA fragments. Siyempre, galing to sa'yo, sa person, sa isang tao. It contains different DNA fragments. And then the goal is that you want to separate and visualize the DNA. Okay, gusto mong malaman yung DNA na nakapaloob or kino-contain itong sample cup na to. So, you will be using gel electrophoresis. So, in your front is an illustration of a setup of your gel electrophoresis. So, to know the DNA, kung ano yung nakapaloob na DNA for your sample, we will be using gel electrophoresis. So, how will it work? For example, meron kang setup ng gel electrophoresis. So, hindi dapat mawala si power supply or power source kasi dito natin makukuha yung current or yung electrical charge na kailangan natin sa principle ng electrophoresis. So, recap lang of your electrophoresis setup. Itong medium na gagamitin natin, as we all know, is a polyacrylamide agarose gel. The texture of this is gel. Gel talaga siya. Parang jelly ang texture nito. Malambot. And then here, located ang inyong aqueous buffer solution. And then nandito ang inyong mga sample wells kung saan doon natin ipopour yung analyte nyo or yung sample na gusto nyo malaman yung DNA. So for example, na-pour mo na yung sample mo dito na nagko-contain ng mga DNA. So dito, nandito yung inyong mga cathode and nandito inyong anode. So once na na-pour nyo na siya, the next step is that DNA will migrate towards your cathode or your anode depending sa electrical charge niya. So what do you think? DNA is a positively charged molecule or negatively charged molecule? Will it migrate toward the anode or will it migrate toward the cathode? Okay, I'll give you time to think. So to answer that question, DNA is a negatively charged backbone. So, paano po yun nangyari, ma'am? So, kung i-recall natin yung structure ng ating DNA, it comprises of three main structures, namely your phosphate, your sugar, and then your nitrogenous base. Your phosphate group is the one that is responsible for the negative charge of your DNA. So, kung mapapansin nyo dito, ito yung structure ng DNA natin, itong mga oxy and ion na to ang responsible sa pagiging negatively charged ng inyong mga DNA. So that being said, your DNA will migrate towards the anode kasi negatively charged siya. So, an opposite attract, so it will migrate towards your anode. Bakit po hindi sa cathode? Kasi it will repel the DNA kasi pareho silang negatively charged. So, ang sigurado natin dyan, it will migrate towards the anode. So since all DNA fragments have the same charge per mass, small fragments of your DNA will migrate towards your anode. And then the migration of your molecules or your DNA through pores is called sieving. So kung babalikan natin yung medium natin dito, meron siyang mga maliliit na pores, as in super tiny na pores that filters yung mga molecules mo or yung mga DNA mo. So the larger the molecules, the harder for it is to pass through. So nahihirapan siyang mag-pass through dito. So kung mas maliit yung inyong mga fragments, mas madali sila makakapag-travel towards the anode. So yun ang principle ni electrophoresis. That's how it works for your molecular biology. So parang pareho lang din siya sa inyong analytical chemistry. Ang difference lang nila, isang pinag-uusapan natin dito is DNA na talaga. Okay, as we said, the smaller the DNA strand, the greater distance it will travel. Mas malayo yung mararating ng mga maliliit. Kasi nga, mas nakakapag-pass through sila dun sa mga pores. Compare dun sa mga malalaki na naiwan sila dun. From the sample well, mas naiwan sila dun sa parang pinaka-starting line nila. We can determine the number of base pairs in the DNA molecule based on the distance. So, katulad ng sinabi natin kanina, kunwari, nandito yung sample well mo. So, naiwan yung mga malalaking molecules. Yung mga malilit na molecules, mas nakapag-travel sila towards the anode. Tapos, i-measure natin sila using the length of DNA fragment base pairs. So, 100 BP yung pinaka-mababa. 1,000 yung pinaka-mataas. Yung mga naiwan na large molecules doon sa huli. So, this is an example of your bands for your gel electrophoresis that has a DNA binding dye. A dye is added to the bands or for your or on your gel electrophoresis para mas ma-visualize natin sila kasi colorless yung gel nyo, colorless din yung mga pinor nyo na solutions. So para mas makita natin siya at ma-visualize natin siya, we need to add binding dye para mag-glow in the dark siya in the UV light. Again, pakitandaan yung UV light. Itong mga to, mga nag-travel na to, nandiyan na po yung mga DNA molecules natin. Each band contains thousands of identical DNA molecules. So, nagkumpul-kumpul na, nagsama-sama na, nagrupo-grupo na yung mga molecules na may magkakapareho ng size. Since all DNA fragments have the same charge per mass, ang mga nagsama-sama dito is yung mga pare-pareho ng size. Okay? And then we have to take note that the shorter the DNA, the molecule, the farther it will travel from the well. So imagine niyo nandito yung well, kung saan tayo nag-start, doon kayo nag-pour, mas malayo yung narating nila, ibig sabihin mas maliit yung DNA or mas shorter yung DNA. Kasi nga po, nakapag-pass through na sila doon sa mga pores. Next is that we can isolate a specific DNA molecule from the mixture via Southern blotting. So ito, isa pa rin tong technique for the molecular biology. It transfers to a filter membrane for further analysis. So as we mentioned, the blotting is a common molecular biology technique that used to specifically identify biomolecules following electrophoresis. So electrophoresis is not a standalone test. We need to make use of the blotting. The molecule of interest is indicated using either a labeled probe complementary strand of nucleic acid or labeled antibody raised against a specific protein. So, ang pinaka-principle lang ni blotting is yung parang gumamit ka ng blotting paper. 'Di ba? Pagka oily na yung mukha mo, ibo-blot mo lang siya doon. So, i-discuss na lang natin yung pinaka-mechanism niya sa mga susunod nating discussion. Pero dito, ang pinaka-overview lang of the blotting technique is that parang gumamit ka lang ng blotting paper to get the molecules that you wanted. Next, another type of common molecular biology techniques is restriction digest, the process of cutting DNA up into smaller fragments using enzymes which only act at a particular genetic sequence. Next, ligation is the process of joining two pieces of DNA together. Ligation is useful when introducing a new piece of DNA into another genome. Next is the more common molecular biology technique. Your polymerase chain reaction is a process used to amplify very small amounts of DNA to amounts which can be used in further experiments. So for example, meron kang sample na sobrang konti na lang and gusto mong maka-extract ng DNA dun sa sample mo. Ang goal ni polymerase chain reaction is to make billion copies of the DNA na nandun sa maliit mong sample. Bakit papadamihin kung, let's say, deadly yung sample mo, tas konti? The goal is to make billions of copy para ma-identify natin kung anong DNA ba yung nakapaloob dun sa small sample na yon. Okay? Yung polymerase chain reaction natin, meron siyang pinaka-tatlong main steps, and that is your denaturation, your annealing, and then your extension. So to discuss this much further, meron tayong separate topic for that. So to give you a quick overview, polymerase chain reaction, we will make billions of copies of the DNA for us to detect kung anong DNA ba yun nandoon sa sample. And in a nutshell, DNA or your polymerase chain reaction is a sensitive process that involves DNA amplification using multi-step of heating and cooling process. So to tell you, this is a sensitive test. Okay, your polymerase chain reaction. Next, going to the history of molecular biology, daanan lang natin yung history ng mga kanununuan natin sa molecular biology. During 17th to 18th century, offspring is from mixture of traits of the parents. It was Gregor Mendel from 1856 to 1863 found the inheritance or the loss of inheritance. Each parent contributes a particle or genetic unit to the offspring. So, na-discover na yon using yung pod. Okay? Parang gulay. Ano ba Tagalog ng pod? It was Gregor Mendel. Next. Excuse me. Rediscovering the Mendelian works, it was Hugo de Vries, Carl Correns, Eric Schermark. Okay, I don't know if I'm pronouncing it right. They discovered the Mendelian works during 1900. The Mendelian law, now accepted based on the nature of chromosome, and then the concept of gamete contain only one allele. So ito, madidiscuss to sa inyong cytogenetics. So it was Thomas Hunt Morgan that discovered the chromosomal theory of inheritance. So using the fruit fly, okay, pinag-aralan niya yung DNA na to at na-discover niya na meron palang chromosomes. In Columbia University, he was a Nobel Prize winner. Okay. Next, the recombination and genetic mapping. It was 1913 when Alfred Sturtevant created the first genetic map. The recombination and genetic mapping, C. Thomas Hunt Morgan pa din, is the exchange of genetic material either between multiple chromosomes or between different regions of the same chromosome. Next, it was Kurt Stern who discovered the chromosomal crossover in the fruit fly. In 1936, the recombination in mitosis producing mosaics. And then Barbara McClintock and Harriet Creighton is the first to produce the genetic map of maize. Next, the early discoveries of DNA. Pakitandaan yung 1869, Friedrich Mischer. Okay? isolated the nuclein. It is the DNA with associated proteins from the cell nuclei. And then it was Phoebe Levine from 1909, 1910, and 1929 discovered this. Next, history pa din po ni molecular biology. Noong 1928, si F. Griffith discovered the transformation of Diplococcus pneumoniae. During 1934, M. Schlesinger demonstrated the bacteriophages are composed of DNA and proteins. So, ang bacteriophage, isa po siyang type ng virus. Even though may word na bacteriophage sa una, it is a type of virus that replicates or invades a bacteria. Do not worry, it doesn't harm humans. And then 1941, it was Bedell and Tatum published the results of biochemical genetics of Neurospora and established one gene, one enzyme hypothesis. During 1944, Avery, MacLeod, and McCarthy identified that the transforming principle of Diplococcus bacteria was DNA. And then 1950, Erwin Chargaff, pagkalilimutan si Erwin Chargaff, demonstrated that in DNA, the number of adenine molecules is equals to thymine and the number of cytosine is equals to guanine. So, sinabi daw ni Erwin Chargaff na ang nitrogenous bases, ito ba, mga nitrogenous bases to of your DNA is equal to 1. 1:1 ng quantity nila. Hence, the Chargaff's rule. Okay, the percentage base pair equality. Ang ratio daw po natin ating mga nitrogenous base is always 1:1. Next, for the history pa din po of your molecular biology, it was during 1953 when James Dewey Watson and Francis Harry Compton proposed the double helical model of DNA based on the studies Maurice Wilkins and Rosalind Franklin. And then they won the Nobel Prize in 1962. 1957, when Mendelssohn and Stahl confirmed the Watson and Crick's semi-conservative model of DNA replication. 1958, it was Kornberg isolated the DNA polymerase from E. coli and got the Nobel Prize in 1959. Next is 1958 when F.H.C. Crick proposed a central dogma of molecular biology. Nakapula, pakitandaan po. And then 1961, Nirenberg and Mathey cracked the genetic present of your mRNA. 1961, Jacob and Monod put forward the operant concept for regulation of gene expression, received the Nobel Prize in 1965, and in 1965, F.H.C. Crick proposed the wobble hypothesis. So the cell is the basic unit of life. Okay? And then it was Robert Hooke who discovered the cell. Okay, it was Matthias Schelling and Theodor Schwann studied the animal and plant cells. And for the cell theory is that, pakitandaan tong tatlo, all living organisms are composed of one or more cells. The cell is the basic unit of structure and organization in organisms, and cells arise from pre-existing cells. So for your reading assignment lang po ito, we have to study the central dogma of your molecular biology. Paki-aral po ng maigi. And that concludes our discussion for today. Thank you.